Hi, everybody. Welcome to today's micro seminar. Our speaker will be Elizabeth McDaniel from the University of Wisconsin in Madison. Elizabeth started out her studies, undergraduate degree in biology at the University of Arkansas. And after completing that degree, she then moved to the University of Wisconsin in Madison. And sorry, I'm getting my own voice in the background. I'll just take that out and just keep talking as it is. So she is currently a PhD candidate in the lab of Catherine McMahon at the University of Wisconsin-Madison, part of their microbiology doctoral training program. And today she'll be talking to us about one of my favorite microbes, Accumulibacter phosphatis from wastewater. But it's not the only topic that Elizabeth's been working on during her PhD. If you're interested, also check out an upcoming paper in M Systems. It's currently in press, I think, on microbial mercury methylation. So she has lots of research interests outside of Accumulibacter. But that's the topic for today, on ecological roles and interactions of these microbes. And I'm really looking forward to your talk, Elizabeth. Over to you.
Okay, thanks, Jillian, for that introduction. Can you see my slides? Are they advancing?
Yes,
okay,
that's fine.
Awesome. So thank you all for being with us virtually for this micro seminar. Thank you to the organizers and Jillian for putting this on. Hope you're all staying safe and healthy. I'm going to be talking about characterizing ecological roles and interactions of Accumulibacter in wastewater systems. So the main things that I'd like for you to take away from this talk today is that wastewater treatment is necessary to prevent downstream eutrophication of receiving waters. Nutrient removal is performed by harnessing specific microbial lineages and their unique metabolic capabilities. And we can use cultivation-independent approaches to get insights into the ecological functions and putative interactions of lineages that are currently uncultivated that are important for nutrient removal. So eutrophication is when there's excess nutrients in freshwater or other bodies of water. This is either due to wastewater effluent or non-point source runoff from surrounding agriculture areas. Eutrophication leads to harmful algal blooms that will take up all of the oxygen. This leads to this really disgusting, really thick algae, and then this can lead to fish kill-offs. And the main nutrients that we're worried about when preventing eutrophication are phosphorus. Usually this is from human or animal waste, and nitrogen, also human and animal waste, but also in the form of fertilizer. And so the way that we design our wastewater treatment systems to handle this is that we take our influent through a series of anaerobic and aerobic tanks and put it through a clarifier to basically remove as much of the waste as possible by sedimentation. And this is a physical process. And what this looks like is you have your waste, and when you let it settle out, this is the part that we want to remove. And so we're releasing this clear effluent that should be removed of any contaminating nutrients. However, we also incorporate concentrated groups of microorganisms into this physical process to help remove soluble contaminants. So the secondary biological treatment that's applied after that primary treatment of sedimentation is referred to as activated sludge. So this is the concentrated group of enrichment cultures of microorganisms that help remove nutrients through their unique metabolic characteristics. And so what we are really interested in the McMahon lab are polyphosphate accumulating organisms, and I'll refer to these as PAOs, that help remove phosphorus from receiving waters. And this is done through a series of anaerobic, aerobic cycles, as I showed before, or also feast famine, where we feed the influent with volatile fatty acids. So let's say acetate. There's sequential anaerobic, aerobic cycles, and this will lead to the removal of phosphorus. So in detail, what this looks like. So let's say we're feeding acetate in the anaerobic cycle. PAOs will take this up and incorporate it into a storage polymer called polyhydroxyalkanoates. This is a high energy containing storage polymer that in the aerobic cycle, when oxygen is available, it uses this for respiratory growth. However, as I just said, this is a really high energy containing molecule. It takes a lot of energy to form this polymer. And so in order to replenish reserves of ATP to form this PHA in the anaerobic cycle, phosphorus is taken up in the aerobic cycle and incorporated as polyphosphate, where these reserves of polyphosphate are broken down in the anaerobic cycle for this ATP. So it's really this handoff between anaerobic, aerobic cycles to form PHA, take up phosphorus, these handoffs. So what this looks like at the cellular level, in the anaerobic phase, feed with acetate, incorporate into PHA. So here's where those reserves of poly P that were made in the previous aerobic phase are used for ATP to store, to make that PHA. And in the aerobic phase, this is where orthophosphate is taken up as chains of polyphosphate. And this process, through sequential anaerobic, aerobic, or feast and famine stages, leads to overall removal of phosphorus and therefore the process of enhanced biological phosphorus removal, or EBPR. There's a lot of acronyms with wastewater and EBPR, so I'll try to keep it simple and not use too many acronyms. And the model PAO that we study in the McDaniel lab is Candidatus Accumulibacter phosphatis. For the remainder of this talk, I'll refer to it just as Accumulibacter. And so this was discovered that when they stained cells and applied with fish, so these dark stains in the middle, these are these polyphosphate granules. So they saw that this new basically microbial lineage that's related to the rhodocyclists in the beta proteobacteria could take up polyphosphate in these sequential anaerobic, aerobic cycles. And so we study in the lab in these lab scale enrichment bioreactors. So it is present in wastewater treatment plants, total probably 10% relative abundance to the total microbial community, which for a full scale complex wastewater treatment plant environment, that's quite a lot. But there still remains no pure culture of Accumulibacter. And so we needed easier ways to study Accumulibacter, to understand its ecology, physiology, metabolic and gene regulation and whatnot. And so we use these lab scale bioreactors that we feed with acetate and provide like an artificial amount of phosphorus that over time, when you seed from a full scale wastewater treatment plant, this is our fearless leader, Trina, literally taking just a cup out of the wastewater treatment plant to seed into these reactors, that over time we can enrich for Accumulibacter and study using molecular and metagenomic techniques. So what this looks like, this is a previous time series of these reactors. And I should say, these are the reactors that we currently have going on right now. We usually have sets of 2. Right now I have a set of 3 that are named after my favorite women in science, Abigail Hellier's, Barbara McClintock, and Cici Martinez Gomez. And so we were actually fortunate to restart these last week as part of our reopening. So I'm excited to get some more experiments going. But what this looks like is, this is an old time series from our first batch of reactors, is that we have a pretty consistent enrichment of Accumulibacter with some other organisms, but in this, the abundance of Accumulibacter can range from about 50 to 80% of the total community abundance. And so in the past, we've used these enrichment bioreactors and genome-resolved metagenomics techniques, which I'll be going over in just a little bit, to understand the functional potential of different populations of Accumulibacter, transcriptional regulation, and make these detailed metabolic models. That would be really difficult to do so from a full-scale wastewater treatment plant, just because that seed community is much more complex than what we can achieve with using enrichment bioreactors. However, so as the the abundance can fluctuate quite a bit. The times where it's like 80% the total abundance. Accumulibacter isn't just one homogeneous population or just one homogeneous species. So we delineate the population structure of Accumulibacter into clades by the polyphosphate kinase gene, so PPK1. So all of Accumulibacter that we can delineate by PPK is mostly 100% identical by 16S. So this microdiversity, sometimes as people recall, we call this of a genus, is really difficult to study with 16S sequencing. So Trina, in her PhD work, used the PPK1 marker, which is important for Accumulibacter's lifestyle. This is where PPK1 is taking these the orthophosphate and forming chains of polyphosphate. And so that's important for the removal of phosphorus. And using the PPK1 sequence signature, we can delineate into different clades. And so we can also use the PPK1 marker to then design specific qPCR primers that are specific to the different clades of Accumulibacter. So this is overlaid with this previous time series. The 16S time series is samples taken every other week. The qPCR time series is once a month. So they roughly match up over time, but they aren't exact for specific dates. But so here when we have basically this constant enrichment of Accumulibacter, this is made up of different clades over time that mostly don't really coexist at one time. It's either one or the other, and there are fluctuations of these clades. And we have noticed that these are our first bioreactor systems. In subsequent systems, it's always this fluctuation of two types of clades. The clades are differentiated into type 1 and type 2. It's usually one is a type 1 member and one is a type 2 member. However, this begs the question, what does this mean functionally? And so as I said before, there are no pure cultures of Accumulibacter yet. So we have to rely on these cultivation independent techniques such as Genome-Resolved Metagenomics. And so how this works is we take our sample from our bioreactor, isolate the microbial DNA, apply this either through high throughput short read sequencing. Now there's lots of genomes that are coming out that incorporate long read sequencing as that technology improves. And we try to take all the short reads that come off the sequencer and reconstruct these into population genomes. Now what this means is you're reconstructing species resolved genomes. So your population genome that might be an Accumulibacter genome is a consensus sequence of related strains, possibly because short read technologies aren't good enough in assembly algorithms to differentiate between those highly similar areas of related strains. So usually your final population genome is a consensus sequence. So we refer to this as a couple different things, such as metagenome assembled genome, or MAG, or population genome, just because it's not one homogeneous strain most of the time. And so using this, we can use metagenomics, assemble these genomes, look at their functional potential and their population dynamics, apply metatranscriptomics to look at their differential expression profiles, really look at their regulation and activity, or metaproteomics, the suite of proteins expressed and looking at functional dynamics. And so there's been a lot of Accumulibacter genomes sequenced to date, not just from our group, but from other groups across the world. And so broadly, which I think is really interesting, is these clade groups that are designated by PPK1 sequence signature. They also fall into these really nice genome groups. So this is an average nucleotide identity, or ANI comparison graph, where it's pairwise. You're just comparing the sequence identity of the whole genome to every other genome in the set. And these are all Accumulibacter genomes. So roughly all the Clade 1A genomes fall into a really nice similar cluster, the Clade 2A, Clade 2C, so on and so forth. And what people have started to do with these sequenced Accumulibacter genomes is then we can look at what are the differences there might be between Clade 1A and Clade 2A. So those were the genomes that we had in our first set of bioreactors. And Jason Flowers, a previous graduate student in the lab, really wanted to look at this. And so some big differences that he found are that in Clade 1A, there are cell wall and capsule differences, differences in cofactor, vitamins, and prosthetic groups. So these kind of auxiliary functions. There's also Clade 1A has a higher amount of really diverse CRISPR regions. And Pancho, whose work I'll be touching on, is working on a separate project looking at CRISPR phage dynamics over time of Clade 1A, hoping that preprint will be out soon, so watch out for that. So as I just said, difference in nitrogen cycling genes, phosphate uptake abilities, and this 1A genome contains a large genomic island with diverse CRISPR regions. And so going back to this, so we can have our population genomes and make our predicted functions of the different MAGs, but sometimes it's really hard to say just from a metabolic reconstruction of putative functions what those genomes are actually doing. Just because it has a gene doesn't mean it's actually performing that function, it just has the potential. And so one way that we can start to answer this is using metatranscriptomics techniques to look at the expressional activity in different samples through space or time of these different genomes and infer putative interactions. So for this first project, that's what I did with Pancho, who was a previous student in the lab, is he performed this nice time series transcriptomics experiment over a normal operating EBPR cycle. So again, acetate is taken up in the anaerobic cycle and is incorporated into PHA, and you have the removal of phosphorus incorporated as polyphosphate in the aerobic cycle. And he found that when he ran this experiment, the bioreactor was mostly enriched in Clade 1A, also in 2C, and a little bit of 2A. And so he was interested in applying genome-resolved metagenomics and transcriptomics to understand what might be the different gene expression programs that these two co-occurring clades are performing over this cycle. So the arrows denote what times along the cycle that he took samples for metatranscriptomic sequencing. So there's 3 from the anaerobic cycle and 4 from the aerobic cycle. And so we were able to assemble 4 high quality Accumulibacter genomes. One we didn't pick up with the PPK qPCR. And so the ones with the stars were the ones we assembled. And by high quality, that usually means by single copy core genes, it's 95% complete and less than 5% redundant. But Pancho also applied long nanopore reads, and 2 of these genomes, 2C and 2F, I believe, are one single contig, and they are closed. So these are really nice genomes that I'm really proud of, and Pancho did a great job with these. And so these in bold are the new genomes, so Clade 1A, 2A, 2C, and 2F. And so we were interested in, since the 2 most abundant clades were 2C and 1A, these are quite different. We wanted to look at their transcriptional profiles. So I'm going to start with just the top differentially expressed genes in each of the genomes. So each row is a different gene in each column. So the first 3 are those first 3 time points of the anaerobic phase, and the last 4 are the time points from the aerobic phase. And so 1A was the most abundant clade in these reactors. And here, the ones I wanted to point out were these genes involved in nitrogen cycling or transformations. So these ammonium transporters, mostly our reactors are configured for phosphorus removal, but they also do have to deal with nitrogen cycling and removing ammonia or transforming ammonia to nitrate effectively. So in Clade 1A, there's this nice warmer colors are induction, colder are repression. And the in the anaerobic phase, these induction of these nitrogen cycling genes. So then 2C is the co-occurring but less abundant clade in these reactors. And what I really want to point out in 2C is the nice profile of these phosphate transport system genes. So the PST ABCS regulon, or this transporter is a ABC type phosphate transport system. So this is a high affinity transporter, where basically if you have an environment that's starved for phosphorus, it's really going to grab onto that phosphorus. And this is important because, going back to our metabolic model at the cellular level, that in the aerobic phase, it's taking up orthophosphate as polyphosphate. So it needs to transport in the aerobic phase that phosphorus and incorporate into polyphosphate. So this nice profile of basically that activity not happening in the anaerobic cycle and induced in the aerobic cycle is just a nice sanity check when you're doing these large scale metatranscriptomics experiments, and sometimes you don't know what to expect. It's nice to see something that you are expecting. And so we can look at these top differentially expressed genes in the different clades, but we also wanted to take a different approach where we wanted to characterize the gene expression dynamics of core and accessory gene content in these two clades. So from that ANI diagram, these genomes aren't really that similar. They're probably similar 80 to 85% by average nucleotide identity. That's a lot of differences. So to characterize this core and accessory gene content, we use an approach that's kind of referred to as pangenomics, where you have your set of genomes. So we have all our Accumulibacter genomes and some outgroups. So as I showed in that tree previously, the Accumulibacter is related to the rhodocyclus and the Thalara. So you have some outgroups that you compare against your set of genomes. And you're basically doing a blast all of these proteins, and you're clustering into different groups so that you can say, of my input genomes, here are all of the core genes, so genes that are present in all of my input genomes. And here are the accessory genes, maybe clusters that are only present in a couple genomes, ones that are present in only one, and so on and so forth. And so we wanted to do this for understanding core and accessory gene content in clades 1A and 2C, and we make these designations where core genes are shared by all Accumulibacter and outgroups. And for performing this comparison analysis, we just use Accumulibacter genomes basically that were assembled from our reactor system. So those are these UW genomes. There's 6 of them, and then we use 2 outgroups. And then Accumulibacter core genes are only in the Accumulibacter references and not the outgroups. So maybe that we can look at some of these Accumulibacter specific gene expression programs. And then the 2C and 1A accessory genes, so genes that are in one clade, but not the other. And you can be exclusionary, where you're only looking at genes that are in that clade and no other clades. Or, for example, looking at accessory genes that are in 2C, maybe a couple of other clades, but not 1A. So I'm only going to show the accessory genes for today because the core genes analysis is still in progress, because comparing core genes across 2 genomes is a little difficult. So this is the Clade 1A accessory expression profile. So as a reminder, Clade 1A is the more abundant clade. And as kind of expected, most of these accessory genes are auxiliary functions. So these amino acid metabolism, CRISPR phage functions, different phosphate transport, things that really aren't central metabolism genes. So before, in those top differentially expressed genes, some of the other profiles that were pretty stark were core central carbon metabolism genes. So we're still looking into these stark profiles, other than Clade 1A does kind of have these nice CRISPR phage profiles. The colors are kind of hard to identify in amino acid metabolism. And surprisingly, Clade 2C also has some nice profiles of CRISPR phage and lipid metabolism. So here's these lipid metabolism profiles where it's induced in the aerobic profiles, in the aerobic conditions, and CRISPR phage kind of repressed in the anaerobic phase. So we're still looking into these auxiliary functions, but basically this is just a different way to look at metatranscriptomics analysis, where you can look at profiles of differentially expressed genes, or we can also look at these core and accessory genes and look at their profiles. And so by the end of this, we hope to compare these and say, okay, here are the core genes that are present in both Accumulibacter, but which ones are actually differentially expressed or expressed differently from the two genomes because they have so many differences. So I want to go back to the makeup of these enrichment bioreactor communities. So we can compare, using these techniques, the main Accumulibacter clades that are present in these bioreactor systems. But as you can see, there's also at times, sometimes abundant flanking community members. And by what I mean by flanking community members are non-Accumulibacter. So these enrichment bioreactors are specifically designed to enrich for Accumulibacter by providing acetate as the sole carbon source and providing artificially high levels of phosphorus. So that's performing the function that we want it to. But we've seeded now 3 rounds of reactors, and anecdotally from others, there's always this persistent flanking community that might not be consistent over time and space. But from the first study that a genome of Accumulibacter was produced, that was based off of our first set of reactors. This study was performed in 2006, and also an Australian bioreactor. So the size of the circle is the abundance of reads that map back to either the assembled genome for Accumulibacter or the assembled contigs of these other flanking community members. And so blue comes from our reactors and red comes from these Australian bioreactors that we were working with a collaborator at the time. And so there's this diverse flanking community that really in these reactors is virtually unexplored. We mostly care about what we enrich for in these reactors, Accumulibacter, because we want to understand EBPR. But so these reactors really, when you seed them, they become this semi-complex community. And as I'll share some data later, there's really only 50 to 60 species in these bioreactors. So this is a really great semi-complex model system to start understanding putative microbial interactions over time. We can really run these reactors with a good amount of control, controlling for the carbon source, the amount of oxygen. So in the lab, we also study freshwater lake microbial communities where there's a lot of unknown factors that are contributing to the environment in these microbial communities, and they're just really complex, thousands of species. And so what I became interested in is how might this flanking community impact what we see about Accumulibacter over time? Is it affecting through these microbial interactions, its gene expression, metabolic functions, and whatnot? Maybe with those fluctuating clade dynamics, is this coming because of something in the flanking community? So again, we can start to answer these questions with genome-resolved metagenomics. So from that previous set of bioreactors from the time series I showed before, we had 3 metagenomes that were collected from the same month that I was able to assemble into 66 high-quality species resolved genomes. So again, these are population genomes. And what high quality means is that most of them are more than 95% complete and less than 5% redundant by single copy core gene standards. And they all have to contain more than 18 tRNA genes. And by high quality standards, they also have to have all 3 ribosomal subunits with short read data. That's really difficult to do because of these really similar areas. The assembly graph just can't deal with that. But 15 out of the 66 do have all 3 ribosomal subunits, which for short read data is pretty good. And so these are the main phyla that the genomes belong to, and the circles are rough ranges of relative abundance. So we can see here from these reactors at this time, they were highly enriched in Accumulibacter 2A. But we also have quite a nice abundance of these other members, so the Bacteroidetes, which I'll touch on in a little bit, because the Bacteroidetes are really quite uncharacterized in wastewater systems. In other systems, we assume here they're the main polymer degraders, because that's what they're doing in cow rumen, the human gut, and whatnot. But we mostly focus on the proteobacteria or nitrospira for lineages that are performing these nutrient removal processes. And so the key nutrient removal and polymer cycling traits among all of these 66 genomes just broadly, as I said, these reactors aren't really configured for denitrification or removal of ammonia. So we're not really enriching for things that have these nitrification, denitrification, or COMAMOX, complete ammonia oxidation genes. Some sulfur cycling, but what I found was really interesting was the presence of these phosphorus transporters and poly P accumulation. PPK is really a core gene that's required for all microbial life to transform phosphorus, but these different transporters like PST-ABC that I talked about before, and PIT is a low affinity phosphorus transporter where if you're saturated with phosphorus, you can take up phosphorus very easily. And the PHA synthesis, so that high energy storage containing molecule that is hallmark to the P removal profile for Accumulibacter, there's quite a few that actually contain PHABC that are required for PHA synthesis, but also degradation. So the Bacteroidetes don't have this specific PHA-D polymerase that degrades PHAs, but they have different ones that I know of. They just aren't shown in this graph. So that's also something that's predicted, that the Bacteroidetes in wastewater and these reactors, they're present in planktonic biofilms or flux, so they're really dense. And that's actually one way that we know that the reactors are really healthy, is they're really nice, dense flux. But it's predicted that maybe the Bacteroidetes are sitting outside of these flux and they're eating the extracellular polysaccharide surface or substance. But we also, we have this predicted functions of some of the key EBPR traits of these microbial lineages, but we also, like I said, transcriptomics can give you a slighter better picture of the actual functional dynamics. And so previously in the lab, Ben Oyserman was a previous PhD student who is now a postdoc that I'm collaborating with. He performed a metatranscriptomic time series, kind of similar to the one that Pancho did, except for he took 3 samples in the anaerobic cycle and 3 in the aerobic cycle. And just to understand Clade 2A, so in these reactors, Clade 2A is the most enriched Accumulibacter at this time. To really understand different EBPR trait expression profiles and have this nice metabolic reconstruction of this clade. And so the really nice thing, if you have metagenomes and a metatranscriptome from the same time, this is a really great case of data reuse. So those 3 metagenomic samples that I assembled the 66 max from came from the same month as this transcriptomic sample. One of them comes from the same day. And since basically assembly algorithms and binning algorithms, the way that we get these mags, have really improved since 2016 when Ben performed this transcriptomic experiment of Accumulibacter, we can reuse that transcriptomic experiment and map that back to all of our assembled genomes to then perform whole community transcriptomics. So there's really quite a range of transcriptional activity. Note that this is on a log scale, because otherwise it would just be Accumulibacter with this really high bar because it's enriched for Clade 2A and everything else is a little low. But I've kind of just highlighted some of the limitations of current metatranscriptomic approaches. They are either gene-centric. So in the first part of this talk, we're applying a gene-centric metatranscriptomic approach where you're looking for profiles of differentially expressed genes to understand expression dynamics over the cycle, or you're comparing certain sets of genes. So you kind of have to know ahead of time, okay, I'm looking at the expression of these important EBPR related functions for phosphorus removal, nitrification, denitrification, or the core versus accessory gene content that we applied in the beginning, or metatranscriptomic approaches are applied in a genome-centric way, where you're reconstructing a specific lineage in detail from these expression profiles. This is extremely manual and really labor intensive. You have to really dig into annotations and these expression profiles, and something like a model like this can take months. So with Ben Oyserman, he's now a postdoc in Wageningen. I'm sorry, I slaughter the name of that every time, in the Netherlands, and with a master's student that he mentored, Doris van Steenbrecht, to create a trait-based comparative transcriptomics pipeline. And so how I'm going to explain this is using the TCA cycle as an example. So the TCA cycle has multiple steps in it, where you're going from oxaloacetate all the way back to malate, or using the glyoxylate shunt from isocitrate, but there's multiple genes that make up the overall trait of the TCA cycle. So if you were using something like a gene-based transcriptomics profile, you would have the differential expression profiles of every single gene. But there are curated trait libraries, such as the KEGG library, that take all of these genes and form it into one module. And so with our pipeline, what we're doing is comparing different genomes that have the TCA cycle, and we're statistically basically measuring the similarity or the difference in expression of a certain trait. So for example, here, the TCA cycle between organisms over time, so that we can say these set of traits are expressed in a similar way, or they are core. So you can have core functions just at the gene presence level, where you say, okay, everything has the TCA cycle, but what does its expression look like? And or they're expressed differently, when we call that niche differentiating. So these amino acid synthesis, vitamin biosynthetic pathways oftentimes are niche differentiating. And what we can do is create this genome-wide network of trait expression. So what this is each square is a genome, each circle is a trait. And what we're showing is basically just the top 15 most expressed, highly expressed organisms from this time series. Since some of them, there's 66 in total, and they have really low counts of expression. So we just took the top 15, and we're looking at basically the overlap, so the middle of these expression profiles over the EBPR cycle, or traits that have these really different profiles from the rest of the community. So apologize for the similarity in colors here, but the brown cluster up here are some of these Bacteroidetes that we hypothesize are the main polymer degraders in this community. The proteobacteria mostly cluster by alpha proteobacteria, gamma beta proteobacteria, but with one kind of interesting exception. Actinobacteria, this is a really cool confirmed polyphosphate accumulating organism that I'll talk about in a second, where basically we haven't colored the different nodes of this, so you'll just have to trust me right now that they're core in central carbon pathways. So everything that's a kind of a hairy mess in the middle are these central carbon pathways, the TCA cycle and glyoxylate shunt, that they're performing kind of the same, which for the glyoxylate shunt, this intuitively makes sense because we're giving these reactors, acetate as the sole carbon source, and to deal with acetate, things have to be moved through the glyoxylate shunt to deal with that. And the main differences, so these nodes that are out by themselves, kind of on the outset, there's a lot of differences in amino acid and vitamin biosynthetic pathways, where many genomes may have the capability to produce different amino acids and vitamins, but it's really costly. So for example, vitamin B12 has a lot of steps and cofactors that go into it. It's a really costly vitamin to make, but it's essential. So if you can streamline that by only a couple organisms in your community making that, and other organisms are hitchhiking off of that, then you don't need everything making vitamin B12. And so the one thing I do want to point out is some things cluster really nicely, but down here is Accumulibacter Clade 2A that clusters with some of the other beta proteobacteria, but Clade 1A clusters like this right next to it is an alpha proteobacteria. So not only are they really different at the genome level, they're only similar by 85% nucleotide identity. We do hypothesize there's some differences, quite a lot of differences in expression level. They are also core in the TCA cycle and glyoxylate shunt, but some of their main differences are vitamin B12 synthesis, those expression profiles, beta oxidation for forming acetyl-CoA, and one of the other most significantly expressed traits that's really different are some of the cytochromes for respiration in the aerobic cycle. So we're still exploring some of these differences, but one trait I do want to highlight is some of these phosphorus transporters, because we were initially interested in making this profile for one reason, because there are PAOs that don't follow this Accumulibacter model, where they take up PHA, where this is formed, the breakdown of polyphosphate, and then use these PHA reserves for respiratory growth. There are confirmed PAOs, so I mentioned Tetrasphaera is in the actinobacteria that removes phosphorus without PHA accumulation. So there are multiple Tetrasphaera strains that have been isolated from a group in Denmark, I believe, and they have been able to use specific experiments. They don't have to use enrichment bioreactors, and can show that they're not uptaking PHA, but still they are forming chains of polyphosphate. And there are some putative PAOs where either based off of genome annotations or expression profiles, where they are predicted to form polyphosphate and cycle phosphorus in this anaerobic phase, but don't take up PHA. And one of these is the Melanobacterial or cyanobacterial, depending on which taxonomy database you're using, this Obscuribacter phosphatis. And it's just one of my favorite names because it's obscure for an organism. But we were interested in looking at basically these profiles of the phosphorus transporter. So just as a reminder, the PST ABCS, this is a ABC type transporter complex for phosphate import. PSTS is the substrate binding component. And this is the high affinity transporter that's used when starved for phosphorus. In these reactors, we are giving an artificially high level of phosphorus. They're not quite starved for phosphorus. And these lines, this isn't normalized expression. It's a way that we're identifying similar profiles. So I don't want you to really pay attention to the magnitude, because the way we've plotted this is counterintuitive. If something is going down, it's actually going up in expression, but mostly that just for each module of the phosphorus, this transporter, they have similar profiles. So their overall pattern is kind of the same. So we have these two attributes or flavors of this high affinity transporter, where we have this set where they're ending, it looks like they're ending low at the cycle, but they're actually ending high, because this plot is counterintuitive, and eventually we will fix that in this pipeline. But for those that have like a high flavor of the phosphorus transporter. This is the network of genomes that are performing that way. And this is Clade 2A. So what was a nice really confirmation of this is we've also assembled 2 tetrasphere genomes in these bioreactors, and one of the tetrasphere genomes kind of behaves in the same way as Accumulibacter 2A with this high affinity phosphorus transporter, as well as some of, I think, Pseudoxanthomonas is a putative PAO. And whereas we have kind of the low flavor type, where Accumulibacter 1A in these reactors is the lower abundant clade, where some of these other organisms aren't transporting phosphorus as highly or based off their expression profiles. But in the future, we also want to look at the PIT transporter, because that's a low affinity transporter, because as I said before, this environment isn't really starved for phosphorus. We are just really saturating it with phosphorus to really get this nice EBPR simulated process. So with that, just going to go over some of the conclusions from these 2 genome-resolved metatranscriptomic studies, that using these methods, we can get some clues into the ecological roles of uncultivated microbial lineages. And using this trait-based comparative omics approach, it might provide a different way for investigating ecosystem processes of a microbial community, since existing metatranscriptomic approaches are either using sets of differentially expressed genes, or you are trying to in detail reconstruct the metabolism of one genome. So we're hoping that this is a more high throughput way to look at a community's expression profiles. And then we'd also like to add in the future more time points and replicants to these studies, because as I said before, these are either like 6 or 7 samples from the same cycle. You can add substrate conditions. We only feed these reactors with the sole carbon source as acetate, but we can also feed different, more heterogeneous mixtures of carbon compounds to compare these functional networks to see how that changes in these ecosystems. So just go over these take-home messages that I hope you got out of from my talk today is that wastewater treatment is necessary to prevent eutrophication, and we do this through nutrient removal of specific microbial lineages, or with our favorite model, PAO Accumulibacter. And because a lot of these organisms from either full-scale wastewater treatment plants or these enrichment bioreactors are yet to be cultivated in pure isolation, we can use these genome-resolved metagenomic techniques to gain insights into their ecological functions and putative interactions. With that, I'd like to thank members of the lab, specifically Pancho we're working together on the first project to compare Accumulibacter clades, Pamela Camijo, who really helped me learn EBPR when I started. Diana, Koti, and Irene are 2 current master students and an undergrad. Are helping me run the current reactor setup, which we don't have data for yet. It's a big undertaking. And then collaborators that are currently in Vonnegut, Ben Oyserman and Joris are really putting together this trait-based comparative omics pipeline. And with that, I'll take any questions.
Great. Thank you very much, Elizabeth. That was really fascinating. It's incredible the kind of detail that you have these days into the function of these organisms. I have just to briefly let you know from the audience, we have one comment, and from Jason Flowers, we have an impressive that was added when you were talking about the mags that you managed to assemble from these metagenome data sets. So yes, very impressive. I'm not sure if you should stop sharing the screen. Maybe we can then okay, we'll be on the screen. I it was very interesting that you can watch these populations over time, the different strains of Accumulibacter. And I mean, you mentioned that they don't tend to co-occur in one batch reactor, at least. And did you see anything from the genomes or the transcription profiles that you would say, this is what I think is the reason that they don't tend to co-occur?
Yeah, so that's a separate project that I've been looking at to kind of understand, do they have something about, their physiology where that's one hypothesis, that they're taking up separate or similar niches and they can't coexist, or at times when they do coexist, it's usually when one of the populations has either crashed for some reason. So what Pancho is looking at is these CRISPR phage dynamics, because there is, Clade 1A has a more diverse set of CRISPR arrays, and so it might be more equipped to handle phage attacks. But then when you can also have, even within these clades, they're not homogeneous populations. We can look at single nucleotide polymorphisms to look at the microdiversity of these clades. That sometimes when you have a clonal expansion, so they are homogeneous, it doesn't leave them as equipped to defend against phage, and you'll have a crash, and that leaves room for the other clade to rise up. So we're still looking into that. There's a lot to consider with the flanking community, microdiversity, phage dynamics, but it's probably the complex set of parameters all working together. Or it's always possible that something just happened with the operation of the reactor at that time, and that's always a possibility.
Yeah, that's true. We have a question from the audience. Firstly, great talk from Yue Li. And the question is, I'm wondering if there's a CP ratio there for acetate and phosphorus substrate addition. I guess the question is asking what the ratio is. I don't remember off the top of my head, other than it's the line of I believe the phosphorus ratio is higher, because if you provide too much acetate, you get bleeding into the aerobic cycle and you'll get things fermenting, and actually the fermenting organisms is what makes this foamy stuff in the bioreactor, and that's usually a sign that you're not getting good phosphorus removal, is when you have these fermenting organisms, and we're providing artificially high levels of phosphorus so that they'll take it up and transform it. So I believe the phosphorus ratio is higher than acetate. Yeah, maybe then a question back to the natural conditions. You mentioned how difficult it is doing metatranscriptomics or community transcriptomics in a complex community like that, but with the high quality genomes in the background for mapping, and if it really does make up 10% of the community, has that been done yet?
There are now, I think in the past year and even this year, now that we can get high quality genomes from these complex systems, I think somebody has performed a transcriptomic experiment over time in a full-scale system, since they got a really abundant Accumulibacter clade. And I know other groups are planning it. It basically is how much money and sequencing depth do you have at your disposal?
Yeah, so that's always the question with transcriptomes, right?
Yes.
True. Okay, another question from Jason Flowers. Have you tried to identify the location of Bacteroidetes in the flock using fish?
So Trina has kind of mentioned historically that when they would use the fish probes for Accumulibacter, there was also a general CFB, I'm blanking on what that stands for, but it includes your Cytophagalus Bacteroidetes, that you would get these little dots on the outside of your flock. Because I remember it was actually last week when I was digging into one of these Bacteroidetes, and how nobody really knows what they're doing, that anecdotally they're kind of on the outside of the flock, and that's where we're getting this hypothesis of they're really chock full of cazymes, glycolytic, these hydrolases, and like the PHA depolymerase. I didn't include this in my talk, but last week, I think, just as a numbers thing, one of these Bacteroidetes that's similar to a strain that was isolated from a full-scale wastewater treatment plant has about 250 enzymes, and the main Accumulibacter only has like 70. So, and I've started to look at that with the transcriptomic profiles to see which ones are the most highly expressed, and it's mostly these PHA degrading enzymes. So, but no, I personally have not looked at where they're located, but I do know anecdotally from Trina that it's been sort of looked at.
And maybe a similar question then to the different Accumulibacter clades. Is it possible to design probes for those, do you think? Are there differences at the 16S that you could use for fish probes? So, I think, they don't tend to co-occur, but, what does that look like? What's the structure like in these real
Right.
plots?
I don't think that's been looked at because the 16S is nearly 100% identical. We just have these there's like general PAO mix probes and general Accumulibacter probes to look at that. But that would be interesting to look at. But yeah, usually the fish probes, you're relying on your ribosomal RNA, and it's all just so identical.
Yeah. And next question from the audience is also back to transcriptomics. Have you seen any transcription factor changes in the transcriptomics?
I haven't really looked into that. We're just looking at the expression of these broad traits, either in central carbon metabolism or these auxiliary traits, such as amino acid and vitamin biosynthesis. But that's usually, we might get into that because that's something we've looked at in Accumulibacter for specific transcription factors.
That might be interesting for those organisms that aren't necessarily closely related, but have similar transcription profiles, or at least expression profiles of different pathways that you showed in the network.
Yeah, that would be something interesting to look at.
Okay, it's been a great seminar. Thank you very much, Elizabeth.
Yes, thank you for having me.
You're welcome. Thanks for preparing this. And thanks to our audience who tuned in from around the world. And stay safe, everybody. See you at the next micro seminar. Okay.